Hello, my name is Brendan Mulvaney and I'm presenting the visual abstract for my research project. It's on understanding the molecular effects of Cebranopadol on brain gene expression with an investigation into how it influences drug-seeking behavior in heroin-dependent rats. I'll begin by explaining important background information. Cebranopadol is an opioid with a unique mechanism of action. Alongside the classical opioid receptors, i.e. mu, kappa, and delta, it interacts with an additional type of receptor, the nociceptin opioid receptor. I'll explain the function of that in detail later. The nucleus accumbens is the area of the brain responsible for mediating goal-directed behavior. It can be thought of as a stop-go junction, and it has been implicated in drug seeking by countless studies. The prelimbic cortex plays a role in cue-associated learning and is also involved in the processing of stress and fear. Cebranopadol treatment resulted in statistically significant reductions in drug-seeking behavior in a dose-dependent manner, as is depicted here. As you can see, however, at the lowest dose, 12.5 micrograms of Cebranopadol per kilogram of body weight, we have a small group of outliers, the non-responders. Since the initial discovery of opium in antiquity, opioids still remain the most effective painkillers known to man. The American Medical Association, however, suggests that of individuals currently addicted to heroin, 45% of them initially became addicted through use of prescribed over-the-counter opioids such as oxycodone. Current opioid cessation treatments such as methadone are effective in preventing withdrawal, but ultimately perpetuate physical dependence. Realistically, we aren't able to stop individuals using and becoming dependent on opioids, so it stands to reason that our next best option is to investigate if opioid-induced damage can be attenuated or even reversed. Long-term opioid use leads to dependence primarily through the induction of behavioral changes, alterations to the endogenous opioid system, and finally, changes in brain gene expression, this being the focus of our investigation. The hypothesis of this experiment is, will treating opioid-dependent rats with Cebranopadol lead to a reduction in drug-seeking behavior related to changes in brain gene expression? These three aims provide structure and direction for testing our hypothesis. Aim number 1 is investigating how Cebranopadol treatment influences gene expression in key areas of the brain, that being the nucleus accumbens and the prelimbic area. Secondly, we want to correlate variance in gene expression among specimens with variance in Cebranopadol's efficacy. This is a comparison between the responder group and the non-responder group. Thirdly, we want to identify and further elucidate the molecular mechanisms by which Cebranopadol reduces the intensity of drug-seeking behavior. The Cebranopadol investigation is occurring alongside a number of sister projects which share a seminal experimental design. To simplify this, rats were allowed to self-administer heroin transfusions via a surgically inserted catheter. They initiated transfusions by pressing a button. A global addiction score was assigned to each rat based on a number of behavioral attributes assessed in later stages of the experiment. These attributes involve escalation of heroin intake during 12-hour access, incremental increases in button presses required to receive heroin, referred to as the progressive ratio, and the strength of the response to heroin-primed drug reinstatement. From this global addiction score, a group of male rats determined to be vulnerable to opioid dependence were chosen. Only this vulnerable subgroup was used to ensure that responsiveness to Cebranopadol treatment is not influenced by differences in predisposition to addiction. Similarly, only male rats were used to ensure sex differences had no impact on results. Those that underwent the 12.5 microgram Cebranopadol treatment were then classified as responders or non-responders. They were classified depending on if there was a significant difference in the intensity of their drug-seeking behavior before and after treatment. Cebranopadol was delivered orally to mimic the most practical delivery in humans. Those that weren't given any Cebranopadol are referred to as naive and act as the control. Once the experiment was complete, the rats were euthanized and the target brain sections were dissected using the Leica CM1900 cryostat set to -20 degrees Celsius. RNA was then isolated using Quagen microRNA kits, and from there a library was created using Illumina-stranded total RNA prep ligation. Once in possession of the data, I began my search with genes that code for opioid, dopamine, and gamma-aminobutyric acid receptors. Pictured here are box and whisker diagrams. Blue represents responders and orange represents non-responders. The log 2 fold value on the y-axis can be thought of as a direct comparison of the intensity of gene expression compared to levels found in naive rats. For example, say gene X had a log 2 fold value of 2.5. That means it is being expressed 2.5 times more than in naive rats. The p-value in this context can be directly related to the consistency of change, with the high p-value shown here indicating inconsistency of change among our specimens. As you can see from this dataset, there is effectively no difference in the level of expression of these genes between responders and non-responders. Let's look at a statistically more relevant set of genes. As you can see, we have a substantial difference in the log 2 fold change values of these 3 genes between responders and non-responders. By focusing on the individual p-values of these genes, we see a stark difference between responders and non-responders. P-values this low suggest that changes in the expression of these genes among responders is highly consistent. A very similar set of results is also found within our other brain area, the prelimbic cortex. You'll recall that Cebranopadol's unique mechanism of action is its agonism of the nociceptin opioid receptor. Discovering that the gene that codes for the nociceptin opioid receptor was highly and consistently upregulated in responders was a very interesting find. Additionally, pre-pro-nociceptin is a precursor to nociceptin, which of course is the endogenous ligand of the NOR. Depicted here is a simplified overview of several functions of the NOR. The NOR influences reductions in rewarding effects of drugs of abuse, attenuation of stress-related behaviors, alterations to pain perception, and anxiety reduction. If we have elevated levels of NOR and its endogenous ligand, it suggests that this receptor is being activated at higher than normal levels. However, these results are still up for interpretation as the NOR is a multifunctional receptor. Delta FOSB is one of the proteins coded for by the FOSB gene, which was shown to be upregulated earlier. It has been described as a molecular switch in addiction and is implicated in drug-related changes to brain structure. It's possible that upregulation of delta FOSB during a period of abstinence from heroin allows for certain drug-seeking behaviors to be unlearned. It's important to note that the mechanisms behind the difference in expression of these genes in responders compared to non-responders remains unknown, i.e., why did these genes become more upregulated in the rats that responded? Before I conclude, I think it's important to highlight some considerations. We had a very small sample size, only 4 naive rats, 4 responders, and 4 non-responders. We also had low p-values, but a majority were not under the statistical threshold of 0.05. This could be affected, however, by an increase in sample size in future experiments. Is it actually scientifically possible to assign an animal an accurate global addiction score and correlate it to alterations in brain gene expression? I think there's too many variables at play. Many components of this experiment and the neuroscience surrounding it have been heavily simplified and condensed for the sake of the audience. There is also an element of speculation regarding the function of these genes in this unnatural context. With the acknowledgement that further investigation is definitely needed, the results of this preliminary experiment show that variation in brain gene expression in the nucleus accumbens and the prelimbic cortex can be correlated with variation in drug-seeking behavior. The strong statistical pattern of these genes, coupled with the relevance of their functions, suggests that it is appropriate to accept our hypothesis, that being, a reduction in drug-seeking behavior is directly contributed to by changes in brain gene expression.